All right, here we go. So, I wanted to go through this little intro for how neutralizing antibodies work. So, this is from a nice little review that's down here, if that's of any use to you. Here's the idea. So, you've got maybe 6 mechanisms that antibodies can work. Now, the most common one, by far the most common one, seems to be method 1. Some of these things, like number 6, only work for one particular virus with one particular antibody, so they're really rare. And it's only a few cases of basically 2 through 6 where we've seen anything like that happening. But the ways it can happen are, number 1, your antibody, which is our little Y here, can get in between the receptor, which is the gray thing, and the virus receptor binding domain, which is the green thing. And it can just block those two from coming together. So that's one way of neutralizing. That's the common way. Number 2 is that the antibody can actually bind to something in the middle, so like a middle step. So the virus will actually bind to the receptor, but the antibody will block it from being able to go to the next step, because these viral receptor binding proteins actually have to change shapes like 15 times. They're like one of those ancient transformers. And so you can get your antibody in there and you can just block one of the steps of the transformation. That's a little rarer that you get those, but it does happen. You can also block it a little bit later. So after it's gone through and tried to fuse with the cell, is like halfway through the process, you can get in there. So 3 is really kind of the same thing as 2, I would say. Let's see, number 4 is you could wait until the entire virus has been pulled inside the cell. So this is one of the things that happens during infection. And you could theoretically hit it at that point while it's actually inside the cell, but before it's actually dumped into the cytoplasm, because the infection hasn't started until the genome of the virus is inside the cytoplasm of the cell. At that point, you're properly infected. And then 5 and 6 only work with a different kind of virus that has a protein shell on the outside instead of a membrane with proteins sticking in it, which is like coronavirus is. So forget about 5 and 6. All right, so here's a nice little chart that shows the relationship of antibodies to neutralizing antibodies. Because you would think, well, if you got 10 antibodies, one's going to be neutralizing, and so if you got 100 antibodies, 10 is going to be neutralizing, something like that. And you would think that everybody that recovers from the virus would make good neutralizing antibodies. You would be wrong, and so would all the scientists who have thought that this is probably how it works, because that's what actually makes sense. So we've got a couple populations here. So the red line is showing you the line that goes through the middle of the data. This is like the average relationship. And we've got 2 different graphs here. We've got total binding antibodies. So this is antibody that sticks and binds in an ELISA type test that looks for any antibody that sticks anywhere onto the spike. And this is a neutralizing antibody titer. So they do a neutralization test that I'm going to show you in a minute exactly what that looks like. Yeah, different sort of thing. Okay, and so you can see we've actually got a couple different populations, or a couple of outliers that I'm going to group up as populations. So these guys over here had high amounts of total antibodies. So you see they're pretty high on this scale, but they're really low on neutralizing antibodies. So they had antibodies, it's just that the antibodies are not neutralizing. In these 3 people, each dot is a person who has recovered from laboratory-confirmed SARS coronavirus 2. Down here at the opposite side over here, we've got people that have very low amounts of total antibody that binds the spike. They have other antibodies. This is the ones that are specific to the virus. But so it's like they made one antibody, but it actually happens to be a decent neutralizing antibody. So that's kind of neat. Those people got very, very lucky, and they might be protected. But people over on this side, they have great antibody responses, but they're not neutralizing. So they are probably not going to be protected from a reinfection with the virus. Yeah, because neutralizing antibodies are how you're going to stop the virus from getting in a second time. And so that's not going to happen. Down here at the bottom, look at this. This is the, the D student immune system population down here. They saw the virus, they had the virus, and yet they somehow beat the virus without using antibodies. There are other cells, there are other ways to potentially beat the virus. That's usually not how it happens. and they probably would not be protected against reinfection, because only neutralizing antibody is going to protect against that. But there must be something in their immune system. Otherwise, I don't know how to explain it. But then look at this cloud up here, this kind of square cloud that we've got up in the top corner. So the line makes it look like, oh yeah, there's a really nice relationship between total antibody and neutralizing antibody. But look, for the same level of total antibody, so right about here, we got this dot and this dot, and you see the difference here? That's 10 to the fourth. This is 10 to the minus 10. So there's a million-fold difference between these 2 points, and super cool, look at this one up here at about 10 to the minus 10. This one over here at about 10 to the minus 3. That is a 10 million-fold difference in the amount of neutralizing antibody, even though they have the same total antibody level. So people that are over here that are less than, I don't know, maybe sort of everybody this side of this arbitrary line is probably going to be able to be reinfected with this virus. And the people to this side of the line are going to be probably not able to be reinfected until their antibody levels decline. And it seems to they seem to decline over the course of maybe 1 to 2 years for this virus. So they have temporary protection on this side of the line and no protection on this side of the line. And the point is, you can't always tell just from counting the total number of antibodies whether somebody is going to be protected or not. Okay, neutralization test. How does it work? So you can be a maniac and you can just do this neutralization test with the real virus and try and put it in the real cells. And in that case, your readout is going to be, if the virus gets in, the cell is going to die and explode. But it takes about 2 to 3 days for that to happen, so it's kind of slow. Or you can get with the modern world, and you can use a thing called a pseudovirus or a pseudotyped virus. You'll see that written in the literature. And in that case, you're using actually the shell of a virus like HIV. You can use the mouse version or actually use HIV. They both work very well. And into that shell, you're going to slot the spike protein. And the cool thing is that it just slots right in, and it works as if this was an intact virus, and they were made to go together. Yeah. HIV just will accept pretty much any kind of viral receptor-binding glycoprotein into it, and it'll work just like normal. So now you've got a thing that looks like the virus, but inside, instead of the HIV genome or the coronavirus genome, you're going to put in a green fluorescent protein is what I put here, or there are other genes you can put in. It's just something that's going to turn a cell a different color for you, because why should life be difficult, right? If your cell can be glowing green and that can tell you that, yeah, this virus got in, that's just so much better than something you have to work harder for. So then over here in the blue circle, we've got our antibody to be tested. And so you do 2 separate runs of this thing. One, you just put this little virus in with cells that Sars could infect. And this spike protein will bind to its receptor and will do its job. Will actually make the HIV shell infect those cells. So the spike protein works kind of on its own. It doesn't need the rest of its virus. And the HIV shell works fine with any old, spike-like protein. It doesn't have to be its own. So that's kind of cool that you can chop and change these parts. So you do that just to prove that, yep, this thing could have got in and this is how many cells it would have hit. And then you mix your antibody with it, and hopefully you do not see as much entry, or maybe you don't see any entry. And so if you have this situation, then you have neutralized. And the neutralization is going to be expressed in an amount. So you're going to dilute your antibody. You cut it in half, then in a quarter, then an eighth, then a 16th, something like that, until you get down to like 1/1,000,000th of the antibody strength. And at that point, you just try all these combinations, all these different concentrations. And at some point, you'll neutralize, and then you'll sort of thin out your antibody enough that it won't neutralize anymore. That's the entire test, but like it says down here, the test is slow, it's not particularly portable, you've got to do it in a lab with some kind of containment, because even a pseudovirus would be considered biosafety level 1 minimum, and maybe biosafety level 2. Yeah, the pseudovirus has to be properly and freshly made. You need some kind of device to count cells that are green and a fluorescence microscope. And yeah, it takes a little bit of skill, and it's pretty expensive, and it takes 2 to 3 days. So this is why people don't normally do a neutralization test, for all these reasons that are here. A lot of people can't do a neutralization test. They don't have the facilities, or they don't know how to do one. Okay, antibodies against SARS. So here's where some of them bind. And so shown down here, these are a bunch of antibodies against SARS. The gray thing is the SARS spike protein. The thing that's sticking up is the receptor binding domain. And if you look, the green patch is the bit that the antibody binds. And they figured this out by actually solving the structure of the antibody with the receptor in every case. And as you can see, it's pretty much just the open conformation of the receptor binding domain. So that one receptor binding domain that is sticking up is pretty much the target for all the neutralizing antibodies. This one over here actually binds to the side of it, and it does not neutralize. So yeah, that's the difference. Here is the receptor binding domain if you wanted to see what one looks like. Here's a little protein ribbon structure down here. This sort of curve right here, like a smile, is right where it binds to the receptor. The receptor has an alpha helix that's kind of curved, and it fits just perfectly right into that little pocket right there. So the receptor binds here. Most of the neutralizing antibodies seem to bind within the gray dotted circle, so they're just directly blocking the thing that wants to stick. That antibody that didn't neutralize and a couple others that don't neutralize sometimes bind to the side of the receptor binding domain or to the bottom of it. And that's fine. That's a sticky antibody, but it's not going to be a neutralizing antibody, most likely, down there. Now, neat thing is that you can actually do an experiment and you can figure out roughly how many antibodies it takes to neutralize a virus. So, yeah, how many licks to get to the center of a Tootsie Pop, or how many, antibodies it takes to neutralize a virus. And so what they did for this test is they figured out, just by doing the test, like, okay, how much antibody do I need to knock out this number of viruses? Great, they've got that number. Then they just mix that amount of viruses with this amount of antibody together and use an electron microscope to take a picture and count up how many antibodies are bound to each virus. So to make it easier to count the antibodies, they're probably attaching a little gold molecule, or rather a little blob of gold molecules to those. And you do that because it really shows up in an electron microscope. It's like a big black dot inside of there. And so here are some of the viruses, and look, can you see the relationship? So here's little antibodies. So for little tiny viruses like this phage MS2, it takes 4 antibodies binding that at the same time is what will neutralize it. For polio, 4 to 5. Papillomavirus, this causes cervical cancer and various other cancers. About 38. Flu virus around 70. For coronavirus, we're just a little bit smaller than a flu virus, so it's going to be somewhere close to 70, but a little bit lower. And for a big old thing like rabies virus, which is shaped like Bullet Bill, that's a pretty cool looking virus, it actually takes 225 of these things. And the neat thing, back on the previous slide, let's go back there for a sec, is that this actually is a nice linear relationship between the surface area of the virus and the number of antibodies that you need. And it ends up with, it also extends to bacteria. So you can neutralize bacteria with antibodies, and it takes a whole bunch of antibodies, something like 1,000, which actually scales up just like all these other things. So that's also pretty interesting. All right, so let's go on here. Here's the math for all you arithmetic fans out there. So here's our surface area. I just picked out the viruses. We've got good pictures of SARS coronavirus 1. SARS coronavirus 2 is indistinguishable from it, essentially. And so we know these are good. This is actually from a paper that I did. So about 72 nanometers, which gives you a surface area of this much, which means that if we assume, so here's the footprint per antibody. So each antibody is going to cover something like 300 square nanometers. That's kind of the average if you take all these and put them together. And so let's just assume that SARS is going to be an average old virus, and so you would need about 53 antibodies per actual virus particle to neutralize. Does that sound like a lot? It sounds like a lot to me. This is why neutralization therapy is difficult. It's hard to get enough antibody in there and have enough antibody run into the right virus at the right time and bind to it in the right way. But on a coronavirus, how many spikes are there? What does 53 neutralizing antibody hits actually look like in terms of the virus? Well, we can answer that too. So this is from that same paper, and what we figured out is that the spikes slot into another protein that's called the M protein, and it makes kind of a little grid, and the spikes can be in the holes between the M proteins. That's where they occupy. And so if you had a fully occupied, 100% perfect coronavirus, which never happens, you would have about 90 spikes on each virion. So that's neat. But you can actually take pictures of coronavirus, and so here are some coronaviruses. This is mouse coronavirus, this is cat coronavirus, this is original flavor SARS coronavirus, and this is a different mouse coronavirus, but we've taken three-dimensional images of it in the electron microscope, which is cool. And these bright white dots here, because I flipped the contrast, so everything that was dark is now light, are little gold particles. So this is what I meant about saying that it's pretty easy to actually count the gold particles anywhere near something, because they are the most visible thing you would ever see. And so your average actual coronavirus, I counted several of these, only has around 79 spikes on it. So 79 out of 90, it's not perfect, but it's pretty good. And so if you assume you got to bind something like 50 and there's something like 90, but some viruses are a little bigger, then you're talking about, well, maybe I've got to occupy about half of the spikes, but if there's only 79 of them and you're putting 50 on there, maybe that's like 2/3 of the spikes. So that's roughly the percentage of the virus surface area that you have to cover. You have to hit, half to 2/3 of those spikes on each virion in order to be able to neutralize. So I thought that was kind of neat way of visualizing it. But there's a complicating factor. Not all virus particles are able to start an infection because they are just very badly made. I mean, the virus puts itself together. So you've got all these assembly defects, you've got mutations, and you've got some viruses that are sitting in the solution, and they could attach to the cell if they ever ran into it, but they just sort of float around in the water, and they don't necessarily ever run into the right protein on the right cell. So yeah. And so there are various estimates. A test that I did back in the day said that about 1 out of 300 SARS viruses was able to infect. More recent test on the mouse coronavirus showed it was about 1 in 10. You can see that's kind of the range that we get for other viruses. For some of these things like, there we go, papillomavirus, it's only 1 out of about 10,000 was actually able to start an infection in that case. For others, it's really efficient, like 1 to 2, but in the sort of 10 to 100 range, that seems about right for viruses. And so instead of needing just 50 viruses, if it's 1 out of 10 that's infectious, you got to cover all 10, because an antibody can't tell the difference between a virus that's going to get in and a virus that's made with all the right proteins, but they're not quite hooked together right, and so it's not going to work. It's like a dud. And so you're going to actually need 500 antibodies, because 50 per virus and times 10 viruses. Or if you need 300 viruses to get one that actually can infect, you're going to need 15,000 individual antibody molecules to bind all 300 of those viruses just to make sure that the one in that group that was going to start the infection is now neutralized. And so this is kind of the math, and this is what's going on behind a neutralization assay. And so I just wanted to make sure that you understood all of this, so that you'd have something you could sort of keep in your mind and think of while we're looking at this next paper. So thanks very much.